Alright, so now in this chapter we're going to learn about tissues. And so again, if you remember, the study of tissues is called histology. And so here we're going to learn about 4 major types of tissues in the body. We have epithelial, connective, muscle, and nervous tissue. And we're going to start with the epithelial. So before we go into that, intercellular junctions, these are the connections between the cells. And so know that tight junctions, these are connections between the cells, and these prevent passage of fluid between the cells. So think about your stomach cells. There's stomach acid inside your stomach. You don't want that acid to seep through your stomach wall, right? So this type of connection prevents that. It could be acid or it could be like another liquid inside of an organ. It prevents that liquid from going in between your cells, from leaving. Then the next one, the desmosomes, know that these prevent tearing of tissues. So these are also connections between the cells, but they prevent the cells from tearing apart from one another, especially if the tissue is in an organ that has a forceful contraction. So for example, your heart cells. Your heart pumps with so much force that you want the cells to stay attached to each other. If they separate from each other, well, then the heart would just also disintegrate. And so here you have the intermediate filaments. And so an example of a desmosome is the intercalated discs for the heart cells. Then the gap junctions, know that these are just the little channels between the cells. So little gaps where molecules can pass through. Okay, so now we're going to start with the epithelial tissue. Epithelial tissue just acts as covering for our body. It lines our cavities. You'll see it in many organs. And so in lab, we've been talking about how the squamous means a flat cell, cuboidal is cube-shaped, columnar tall. And for the layers, simple means one layer of cells, stratified more than one. Pseudostratified was the fake one, and this appears layered, but it's not. Okay, so this chapter is just really just memorizing the functions, locations for these. And so I'm gonna show here what I want you guys to know. For this one, the function, know that this is for diffusion of, it could be gases, molecules. So because the cells are so flat, so skinny, molecules are easily able to go through. And know that this is a good example for the location, the air sacs or the alveoli in our lungs. Next one, the simple cuboidal. So now the cells are more cube-shaped. This is for secretion and absorption. You're going to learn more about this in A&P2 when you learn about the kidneys. But so know that a good example of the location is the kidneys for this. Simple columnar, so now the cells are longer. Some may have microvilli. Microvilli are these very short extensions on the cell surface, and it's for absorbing nutrients. And so again, know that secretion and absorption is the main part. And for the location, let's just write down our digestive tract. Stratified squamous, so here many cells are on top of each other, and the cells at the top are flat cells at the top. Having many cells on top of each other acts as protection for the deeper cells. So that's the function for this tissue. And for the location, we can write here skin. Let's also know these inside our mouth, vagina, anal canal. Now, there are also stratified cuboidal, stratified columnar. We're skipping those. They're very rare in the body. But just so you know, we'll see those in a bit. Pseudostratified. Here, it looks like many layers, but it's actually a single layer because the cells have these like weird shapes, kind of like triangular. This tissue has cilia and the goblet cells. So cilia are longer than the microvilli. Microvilli are for absorbing nutrients, but cilia are for moving substances or mucus This is in our respiratory tract. Protection from infection could be a good function for this. Also, moving mucus. And we'll say for the location, trachea, our windpipe, has this tissue. Transitional. So now this is also many layers, but it's different than the stratified squamous because the cells at the top are rounder. So they're more dome-shaped, more oval-shaped. Now, when the bladder is stretched, the cells actually do get flattened. They stretch as well. But so for this one, function for transitional is that it stretches. And we'll say for the location, the bladder. That's a good example for that. All right, epithelial tissue also makes up our glands. These are actually the simple cuboidal, simple columnar tissues making these up. So again, don't worry too much about those. I'm not going to ask those on the lab exam or anything, or the lecture exam. But here, the important thing on this slide is that know that there are two types of glands, endocrine versus exocrine. Endocrine glands secrete their products, whatever that they make, into blood vessels or lymph vessels. And the exocrine glands secrete their products into ducts. Okay, so usually glands are either endocrine or exocrine. Pancreas is an exception. Pancreas is both endocrine and exocrine because pancreas makes insulin, glucagon. Those are the two hormones that it makes. Then those hormones are put into the blood vessels that are around the pancreas. So because those products are sent to the blood vessels, that's called its endocrine function. Okay, so I'll write down here endocrine into the blood. But our pancreas also makes digestive enzymes, which are going to go to the intestine. And so then the other parts in the pancreas making those enzymes then send the product into this green tube that you're seeing here. So that's called a pancreatic duct. So then because those products go inside the duct, that's called its exocrine function. Okay, then the next thing, types of glandular secretion. So what we need to know is, here it says merocrine. This type of releasing the product from the cells is using exocytosis. So if you remember, exocytosis just meant when the products are put into these vesicles, these circular shapes, then these vesicles go up to the surface and they empty out their products. So that was exocytosis. Okay, so know that merocrine is exocytosis. Sweat glands use this method. Then an apocrine type of secretion, and this is used by apocrine sweat glands. So here, again, the stuff is put into the vesicles, but the top portion of the cell breaks off. It also breaks into pieces, and whatever was inside, then now is outside, comes out. So for this, let's underline top portion of the cell is shed. Okay, and the last one, holocrine secretion. So here, know that the entire cell explodes. It ruptures. So then whatever was inside comes out. Okay, so an example of this is the sebaceous glands that we're going to learn in the next chapter. Okay, so for now, just know the definitions. What is merocrine? That uses exocytosis. Apocrine, top part is shed. Holocrine, the entire cell ruptures.